Michael, please go ahead.
Okay, sounds good. Thank you so much for having me. Okay, so I'm going to talk about our efforts over the past decade or so to try to think in a coherent way about living synthetic matter. And what that means is basically the question of sort of what would it take to make soft matter physics, right, in some way emulate something that had biological function that was alive, whatever that means, in some form. And this is something that I've been talking about now for an embarrassingly long time, and essentially we haven't really gotten very far yet. And so, so anyway, that's where this thing is going to end. But of course, like in all stories that end in a sad way, there's lots to say. Maybe there's even more to say when it ends in a sad way. So what I'm gonna do. My plan today is to sort of get and sort of tell you about our where we are, what we've been thinking about, how we've been thinking about it. And I would love just whatever kind of open discussion everybody wants. So I was told that I should prepare this as 3 20-minute sections, and this is supposed to be pedagogical and for students. So I'm going to do my best to do that. And my 3 20-minute sections, actually I should look at my clock so that I don't violate this, are the following. So the first section is basically going to be the introduction. I'm going to sort of discuss what the question is, at least the way we formulated the question. You, of course, can criticize that. You can criticize any aspect of this, but that's what I'm going to do in the first 20-minute section. Then the second 20-minute section is going to be about some model problems that we have thought about over the years for how one could make a soft matter system, start to address the question of synthetic living matter. And that I sort of called statistical mechanics plus because these are basically nice problems in statistical mechanics at one level, but with the hope was to formulate them to get us to something that was living synthetic matter. And then finally, I'm going to talk about basically our ongoing attempts. This is the third section. I'm going to talk, we have a bunch of ongoing efforts that are sort of in flight right now, that are aiming to try to instantiate some of these ideas with actual materials. And that's really where we're going with this. And there are sort of different levels of sophistication. There are colloids, which I expect many people here know well, and that sort of have, for a long time, was always where we were going at with this, which is sort of the topic of the second thing in a way. But the three, is work that Chrissy Du, who's a postdoc at Harvard with Paul McEwen and Itai Cohen, who have an amazing, what I think is an amazing experimental system that they call experimental handshake materials. And I'll tell you a little bit about that, just more about the system than anything. We've been working with David Baker's lab for a while, trying to help design protein self-assembly, and we're not very good at it, but we're still trying. I'll tell you about that. And I'll tell you briefly about a project which I at least view in the same universality class with Wendell Lim, who programs cells to basically do things like this. And that's going to be the third part. So I just spent 4 minutes. I don't know which of the 20 minutes that comes out of to sort of introduce this. And so that's where I'm going. So I'm going to now do the first 20-minute section. And I should say, I guess maybe we're all used to this now of speaking to our screen and hoping that people think it makes sense. I just tried this morning with about 50 graduate students. This feels quite similar. If anyone has anything to say at any moment, please just say it. I don't know, David, maybe you have other rules, but I'm self-conscious talking to my computer.
Sure, so, that sounds good.
Okay, so introduction. So this is the sort of usual nonsense. So the usual nonsense the way it goes is this: what would it take to create to create synthetic materials with the properties of living ones? And there's the obligatory picture that you're supposed to put up, which says, that humans were sort of dumb. We have to place things where they go, whereas biology is smart, right? Things self-assemble into functional components, and you look at biology, and there are all these examples. Like there was a graduate student, Nick Kuchia, who's been working with Zvonimir Dojic at Santa Barbara, and yesterday he gave in our group meeting a wonderful talk about the bacterial flagellum and all of its magic, which we all know. But there's the nuclear pore complex. There's just all of these machines that self-assemble and do things that we can't create. And there's the sort of question of what would it take for us to be able to design things that did this. And of course, it's even more complicated in biology because everything has to happen in a soup with lots of other stuff going around. And so your components basically can't interact with each other, because if they do, they presumably degrade performance. But somehow the system is designed in such a way that it works anyway. And we're not nearly that good, even though we've gotten much better at designing components. And so the question is sort of what would it take to sort of make actual progress on this? And and even why, like, what is the difference? And so this is sort of, this slide has a lot of words in it, but it's basically a hypothesis. So there's the whole slide is a hypothesis for what the difference is. So nature creates soft materials which are far more intricate and dynamic than we do. And the question is why? And one hypothesis is that nature has programmable protocols for creating building blocks, and it has had, billions of years to basically, fine tune the parameter sets for those said building blocks. So, right, there's a school of thought that says that the reason why it's so hard to do any of this stuff is because you have to tune it so specifically for it to work that, it might be robust. There might be a wide range of parameters or whatever such that the thing works, but it's, but still you have to find where you are in the parameter space for it to work, and that's tough, and that the program building blocks sort of far exceed the functionality of a random set, and so and then on top of that, natural systems are apparently, I don't know if this is true, but sort of seems reasonable that they're reliable and fault tolerant. There are actually or error correction mechanisms built in. What would it take for us to sort of have these sorts of features in synthetic properties? And so and I guess and the question is that you can sort of, and I'm going to sort of mention this in a minute, is that this is the sort of thing you can do in a computer model if you want, but like I don't care about computer models. Like we need to do things in such a way that we can build them that they such that they respect the laws of physics. And the hypothesis that we've been following on this for a while, and like I said, this is sort of roughly unsuccessful. I was going to say marginally successful, but maybe that's an overestimate, is that maybe one could get a good bit of the way there with material components that interact via local rules that are coded by short range heterogeneous interactions. So short range meaning that, imagine you have a bunch of building blocks and that like they stick to each other with specific strengths, and imagine that you could code this matrix in some way. That is, the question is, what is that? How far could you get with just that? Now, of course, if that's all you had, that's not enough because you need some way of dumping energy in the system and things like that. But at least that's a start to turn this into a sort of physics problem. So now, so of course, the link between simple rules, that is, simple rules like of the sort of like a binding energy matrix or something that looks like this, and complex behavior is sort of old. Right, this is not this has been talked about for a long time, and the sort of first paper, I mean, maybe there are earlier papers, but maybe the first good paper that sort of discussed this was von Neumann's lectures that you can find online on which are called the theory of self-reproducing automata. And in these lectures, I mean, von Neumann was trying to answer what was really a logical problem, namely, is it possible to make mathematical rules that can one make a mathematical model that basically instantiates complex behavior? And as in order to do that, he invented the cellular automata, and he wanted to basically have a cellular automata that copied itself, right? That you just put a seed down and the seed copied itself and made a self-replicator. And in order to do that, the simplest thing he could think of was a triangular lattice. A cellular automata was a triangular lattice, and it had 29 states at each lattice site. And he then needed to define transition rules for how at every step of the automaton, each of the states would update itself, giving the states of its neighbors. And this paper is really a tour de force. I mean, calling von Neumann a tour de force is almost trite. It's so true, but still, the amount of detail that he went through to sort of design the rules that were needed, the automata rules to make this happen, is really just extraordinary. And this was the simplest system that he was able to find. And I mean, the way he writes, I guess I find inspiring. I sort of reread this a couple of weeks ago for some reason, and I just he talks like he's talking about physics, like he's working on our problem. He says, he says, imagine that he says, draw up an unambiguously defined elementary parts list Imagine that there's an unlimited supply of these parts floating around in a large container. Then one can imagine an automaton functioning the following way. It's also floating around and it picks up the parts, puts them together, or if aggregates are found, it takes them apart. And this is sort of like what we're talking about. I mean, he says, by the way, this is an algorithmic description of what an organism does. But, the question is can we, many of us work on things that are floating around in containers. And so surely we can figure out how to design them so they do this. And he then goes on to say, by axiomatizing automata in this manner, one has thrown half the problem out the window, and it may be the most important half. One has resigned oneself not to explain how the parts are made up of real things, specifically how these parts are made up of elementary particles or higher order chemical molecules. At some point in this, he says that here, one does not ask the most intriguing, exciting, and important questions of why the molecules or aggregates which occur in nature really occur in these parts, are the sorts of things they are. And then he says, this is a peculiar range for an elementary object, since even on a linear scale, at least 5 powers of 10 away from really elementary entities. So this sentence is fascinating. He's sort of remarking that there's 5 orders of magnitude between 10 microns and an angstrom, whereas so the machines are operating at a larger scale, and why and how, what does this mean from the point of view of physics? And so anyway, this is I at least find this inspirational. It's a
So Michael, when did he do this work?
when did he do this? So von Neumann, this was the end. The date on this was the end. Oh, David, you came. It's nice to see you. So that's so von Neumann. The date on this is, I don't have the date. This is the early. I want to say 19. It was the last set of lectures. He gave these lectures at the University of Illinois, and they were and they were compiled by Arthur Burks. And I want to say it was right before he died, which was maybe the early 50s. I think that's right. I don't know, maybe someone else who's more scholarly knows, but it's something like that. This was
Thanks.
So in any case, so this field, the field of cellular automata, has sort of, I mean, I'm not sure who I'm going to offend by saying this. I'm sorry. I guess you could always just turn off the computer. So it's gone to hell ever since, basically, because, John Conway came up with this brilliant idea that he said, actually, this was not an example of that last sentence. I have to be careful of what I'm saying. That is, John Conway came up with this thing, can I simplify? Can I build, a cellular automata with mathematical rules that is simpler than what von Neumann invented? And he invented the game of life, which really only has 3 rules. And, with the 3 rules, was able to, find, large numbers of interesting behaviors and within the game of life. And this is like a community that persists to this day, of people who sort of study just all of the remarkable structures that can be made. There are oscillators, there are what are called catalysts. This is a glider gun in which you start out with an initial structure and it makes more structures, which then comes back to the initial structure. And then there are what are called breeders, where stuff emits even more stuff, which then even emits more stuff. So this is sort of quadratic growth of stuff. And, there's just sort of like an infinite amount of richness in this game, which is given by simple rules. Now, these rules that Conway programmed, of course, require, if you were going to instantiate them into something physical, they require energy to make. And so it's sort of the question of what this means in terms of the physical world is an interesting one. But one question which I sort of discovered recently that has been an open problem in this community for a long time was sort of von Neumann's, right? Which was that's really nice that the game of life leads to all of these patterns, and but does there exist an initial condition that leads to a self-replicator in the spirit of von Neumann? And this problem was unsolved by 2008 until 2018. And these are the essentially blog posts on the Game of Life blog where people were talking about it and finally figured it out. And this essentially required finding an initial condition which was able to copy itself exactly, so that at a certain time you only on the board had a copy of the original thing. So I guess my thought is that von Neumann's thing was complicated. He required 27 states for each lattice site, and he had to get these crazy transition rules. This is one with simpler transition rules, but one is not let off for free here either. One has to, if you want a self-replicator or any particular behavior, you have to search for the initial condition, and how do you do that? And so that's what this is. And so I guess the sort of getting back to lessons, I mean, the models, both models have two components. There are nearest neighbor transition rules, there are initial seeds. Both are basically arbitrary. So the reason I made that negative comment about cellular automata is because the problem that I've always had with this is that, but it has nothing to do with physics or the real world. So what am I supposed to do with this? I mean, that is the fact that I can make a mathematical model that breeds such complexity is interesting, perhaps because it shows that logically mathematical rules can encode for this, but it doesn't actually help me in solving the problem that I want to solve. And that's the problem. And there's a price either way. You either have complicated rules or you have to find an initial condition. Either way, even in this world of simple models, this thing is hard. And I actually don't think that this sort of anecdote, the von Neumann game of life anecdote, should make one feel more confident about the question, about being able to sort out the question that I presented at the beginning, namely, can we sort of invent materials within which we build rules through the ways that they interact to do interesting things? Because if it's that hard in a two-dimensional lattice model, then good luck with reality. But anyway, that's the sort of question that this raises. So okay, so let's see. So how are we going to do this? So that was a sort of supposed to be motivational. So I mean, you could imagine there's this sort of old field of self-assembly where what you do is you take a bunch of blocks, they could be identical blocks, you put them in solution and you let them assemble. And of course, what happens is that given the energy and the entropy of the blocks, you can compute the probability distribution of what happens. And then, and the thing about this, of course, is you get whatever you get. You can't, you're not allowed to choose what you get. You get what you get given what you've put in. So the only way that you can sort of program materials to do things is if you can put information in somehow. And of course, there are various ways of doing that, but simple way to do that is to think about making blocks which stick to each other in different ways, and having lots of blocks to create what one wants. Now that is itself not enough, but at least it's a start. And this sort of made us to start to think about what we call the heterogeneous limit. So the heterogeneous limit, so the homogeneous limit is sort of physics that we sort of, at least I learned when I was younger, namely that we have many copies of the same thing, and we learn how to compute the interactions of the many copies of the same thing. But then there's the sort of other limit, which is really, I think, best typified by the DNA nanotechnology community, where basically everything is different, and so you have to compute in that limit. And so so for a while we sort of amused ourselves, and this is sort of part 2 of the talk, with sort of mathematical models of this second category, trying to see if we could get them to find parameter regimes where they would do things that are interesting. And that's what we're trying to do. And I guess this slide makes the comment that there's been really a lot of progress with programming heterogeneity over the years. I mean, there's sort of coding with DNA, there's coding with shapes. This is a very nice review article by Sharon Glotzer and Mike Solomon, sort of talking about all the shapes that can be made in conjunction with depletion forces. One can do a lot. And I mean, you could even this sort of our interest in sort of David Baker's systems is just that, I mean, this is sort of cheating to try to do this out of proteins, because proteins, I mean, that's not exactly not living. But I mean, on the other hand, if they're not doing anything, they're not living. So, and if you can program them, then terrific. So maybe there's something that can be done there. And let's see. So you all know about this. I'm now at 20 minutes, so I'm going to try to finish part 1. So that there's been tremendous work over the years on particles coded with DNA. I mean, so so in the soft condensed matter community, there's John Crocker, Gang, Paul Chake, and Dave Pine. There's been tremendous work. There's also Chad Merkin for a long time has been coding nanoparticles with DNA and showing that by choosing the strands well, that he could make these sort of interesting phase diagrams of different materials. And this paper actually is still only a small number of types, and there's sort of a lot of room to do more. So finally, the last example I want to show you, and I'll come back to this at the end. This is something that we've been fascinated by lately, and it's also sort of cheating, but I think it's cheating in an interesting way. So this is an experiment that Wendell Lim published a couple of years ago, in which what they did was they considered cells developing organoids, essentially fibroblast cells, in which they re-engineered the notch pathway. So we used the modular SynNotch pathway that basically had the feature that when cells, they programmed it so that when cells contact each other, the cells would then start producing some genetic program that they could control. And what they chose to control in this paper, which I think is really quite interesting, is to control the expression of coherence. So when cells contacted each other, they would start expressing coherence, and thus you could regulate their adhesion from contacts. So this is a sort of interesting kind of program. It's different than what you could do with, colloids or something that was dead. You could sort of have particles which, as they interact with each other, they change the way they interact with each other. And that seems like an interesting system to program. And so this is another experimental system that at least we've gotten interested in. And finally, just as a last thing before I stop, in this context, and I apologize for all of these cellular automata, I must tell you, if you had told me 10 years ago I was going to give a talk with cellular automata in it, I would have cried. So I'm not sure what that means. But in any case,
Michael, could I take you back to the previous slide for
please,
a moment?
Please.
Yes. So I'm intrigued by this the
Sorry,
previous slide.
Yeah, no, I'm trying to go back. Sorry, I'm having trouble.
Sorry.
Oh, here. Go. Yeah.
Yeah. So this notion of once they interact, then they're in some sense activated to do something. They're living in that sense. Is this a sort of an active matter version of self-organization?
I think you could call it that. I mean, Wendell would say he's programming, well, he says he's programming self-organizing multicellular structures. So he's using the fact that cells are themselves little computers. And, if you can only make it so that when they do something, that they can produce something, then, he can program it. And there's like tons of stuff that he can do. I mean, you can program cells so that when they bind to each other, they excrete morphogens. You could program cells such that when they detect the morphogens, they divide. You could program cells such that, in this case, in this paper, he programmed them so that when they contact, then they express coherence, so they change their adhesive properties. And I just, from the point of view of the picture that I pointed out earlier on, which is that you have particles that stick to each other specifically, coherence stick to each other specifically. So now we have specific adhesion in a system that, has memory in a certain sense. And so I just think, from the point of view of trying to figure out how to design complicated things, this seems interesting. I don't know if
very much so. Thanks.
Yeah. So okay. So this is a sort of I don't want to call it this is the cellular automata version of that, which I actually find fascinating in my newfound fascination of cellular automata. So remember, I told you that cellular automata, right? There are rules, and you program the rules. The rules are of course fake. They but then they do these things, and then you can ask, well, can I do that with real things? So the analog that I would put to Wendell's system is this one. So this is a paper or a blog post, because I guess now people write blog posts, they don't write papers, that was written by Mike Levin, who's a developmental biologist at Tufts. And what he did was, so he's interested in planaria development. So, you cut off the tail and the tail regrows, and well, where was the information that told the tail to regrow? And so what he decided to do was to see if he could instantiate that in cellular automata. And so he did something that von Neumann couldn't have done, which is that he replaced the cellular automata update rules with a neural network which learned them, which had the feature that he was able to consider, a dramatically larger set of update rules than you ordinarily could. And he was able to make cellular automata. I can give you the link to this. The link is here, but unfortunately you guys don't see it. It's actually quite amusing. He was able to make cellular automata, which when he damaged things, they regrew. So these are all different instantiations of the same thing. And these are, again, this is like in the spirit of von Neumann, right? They grow into something, and then when you damage it, it regrows. So like the butterfly comes back. I mean, there are errors, but it still certainly comes back. But he did this only through local rules. There's sort of local rules that are acting all the time. There's nothing more than that. And I don't know, I just find this interesting. So, okay, I think that might be the end. Design space is large. I said that. Accessible solutions, blah, blah, blah. Okay, that was my first 20-minute section. So I'm stopping now because I was told I should stop after 20 minutes.
Think of it as loose instructions.
Well, I could keep going.
Maybe Michael, I could sort of follow up on that remark. So if this type of self-organization with agents, when they interact an interface, then do something, is there an upscaled version of that where you could think of an ordering as a mean field continuum version of that with interfaces which are...
Well, wait, sorry, see, I didn't hear exactly. So a mean field version of...
So again, sort of imagine, again, you have an ensemble of these agents now.
Yeah.
And then you're zooming out to see sort of the large scales structures that might emerge. Is it an interesting interface dynamics that might?
Yeah, so I'm sure you could. So, the thing is, so I have very mixed feelings about this whole thing. So I'm sure that you could do this. You could write down, interface dynamics of. I mean, they're discrete rules. So clearly, you could coarse grain and make interface dynamics. So my hesitation about this is that, and really my hesitation about whether this is actually even interesting to study at all. I think it's interesting as examples because it sort of shows what you can do with rules, like in the spirit of von Neumann. But what we really need to do is make things that are real, right? Like clearly this shows that one can get behaviors like this when they're properly programmed. But the question is not, so the question is now not, can one program to get these behaviors? The question is, can we please do it already?
Well, I was going to say that one of the models that came to mind, which is real, is the formation of leaf shapes. So the shape of a leaf is in some sense a sort of signal transduction sort of process, which I thought that was reminiscent of.
Yeah, but the problem with leaf shape, I agree. But the thing about leaf shape is, okay, so what question do you want to answer? If you want to understand leaf shape, then you need to understand the cellular programs in the cells that are making up the leaf, right? The fact that I can emulate that is interesting. Like if I couldn't, then that would also be interesting. But I'm not sure there's anything more to say about it once you've once you've done that. Does that make sense?
Yeah, sure.
Oh, somebody said, Manuel said the paper on automata was published in 1966. That's when the paper was published, Manuel. But he gave the lectures much earlier because Burke had to write the lectures. There's a long introduction from Burke who describes how he transcribed the lectures. And von Neumann, anyway, so. Other questions or comments? I can keep going. No? So should I, David, do I just keep going?
Yeah, sure.
Oh, somebody asked, for natural self-assembly, what comes first? Structure or function? Good question. I have no idea. It's a good question, though. Okay, so second 20-minute section, model problems with self-assembly. So we sort of started working on this, I mean, now more than a decade ago with Vinny Manoharan, who was interested in, with Guangdong Meng, colloidal self-assembly. And this is a situation where, right, I mean, what Vinny did and what Guangdong did was to put colloids in a box to let them interact with depletion interactions, and they would then form, clusters out. And we sort of found ourselves in the business of computing, right, what one should find from statistical mechanics. These are nice exercises in statistical mechanics, but given that the objects have no information in them, the complexity is dwarfed by what I was just talking about. On the other hand, in the spirit, I know this is sort of a school, actually, I don't know what this is. So I just, in the spirit of whatever, I have statistical mechanics on one slide. This is one slide. Actually, David is here. I'm going to embarrass myself in front of David. This is statistical mechanics on one slide. So I don't know if that's right. So basically the objects basically obey, in this case anyway, these are not quantum objects, these are classical objects. They obey a classical Langevin equation. The noise strength is set by the fluctuation dissipation relationship, and there is a Fokker-Planck equation that's associated with the Langevin equation that basically gives the dynamics of the probability distribution of the objects. And one can, of course, study the dynamics of this, or one can look at the equilibrium solution. And the equilibrium solution basically is just given by this, where C is 1 over the partition function. The partition function is just given by this integral where U of X is the potential, which again, as I said, in the cases that we're considering are sort of short range-ish. And then the partition function sort of has 3 parts, or 2 parts. There's the sort of energetic part, which is like whatever the binding energy is in the system that you're looking at. And then there's the entropic part. And the entropic part basically consists of partition functions that can be derived directly from from reductions of this integral. And so that's the sort of, if you want to sort of be in the business of predicting yields of things, that's what you have to do. Of course, the designability of these terms is, well, these terms are not so designable. I mean, the vibrational modes, I guess you could design. The what sticks to what you can design, and the rotational partition function is whatever you get. And at least in these colloids, and this is again sort of from a long time ago, what we found was that there were sort of differences between the different the different clusters, the yields of the different clusters that can form. So this is comparing for 6 particles, the 2 structures of 6 particles. One is octahedral and the other is polytetrahedral. And the octahedral one was strongly suppressed over the tetrahedral one, even though they have the same number of bonds. And this was an entropic effect that had to do with the rotational partition function. And we somehow spent some period of time computing these things. This is for 7 particles. Again, all of these structures have the same number of bonds, but there's some variation in their yields that comes from entropic considerations. And these are sort of nice problems. So that's sort of homogeneous assembly, of colloidal clusters where everyone is the same. Now, there's a sort of analogous problem for homopolymer assembly. So what we could do is the following. That what I just talked about was finite number of objects. You also could put objects in solution at a fixed concentration. So if you make the concentration C, then you can sort of introduce a chemical potential, which is a Lagrange multiplier that enforces that, which is related to C through e to the beta mu. And you then can compute partition functions to sort of figure out what happens if you have homopolymers. So by homopolymers, what I mean is I have little monomers that stick to each other that will make long polymers. And the question is what happens? And the answer is there are sort of 2 possible things that can happen in this model. One is that if the binding energy is smaller than the chemical potential, namely that the monomers are happier in solution on their own than sticking to each other, then you will get a distribution of yield of structures that decays with the length of the polymer. Whereas in the opposite case, where the binding energy is bigger than the chemical potential, you will get the opposite thing. And so, and you can sort of, and that's basically what happens in this homopolymer assembly problem. Now, those are the sort of baseline problems, as it were. I mean, what one really would like to do is to program the structures to form non-trivial things. And so, right, if we're fixing the number of parts, so we're not doing finite concentration, if we're just going to fix the number of parts, then, a sort of simple thing you could do, and this is so simple as to be, well, completely naive and dumb, basically, is you could say that if you want to build a structure of, at some point we were interested with Zuravchik of building Big Ben, just can we build out of colloids that were coated with specific interactions anything? And for some reason we took that to be Big Ben. Actually, well, it doesn't matter why. So this is the adjacency matrix of Big Ben. This is a Big Ben that's made out of something like 65 particles, and this is the binding pattern that you would expect of the particles with each other, so that if it formed exactly the structure. And then what you can do is you can basically say that what I'm going to do is I'm going to use specific bonds that make every bond only stick to the one that it wants to, and everybody else I'm going to make the energy much less favorable. And so that, of course, being able to do this requires lots of specific interactions, but you could still ask, if you did that, what yield would you get? And these are yield curves for a bunch of different structures that were designed this way. This is yield as a function of temperature. Of course, at low temperature, the yield is low because the system gets stuck. The yield is low because the system gets stuck in kinetic traps. There's like a glassy type of transition at low temperatures. And then when you're at very high temperatures, the whole thing falls apart. And there's this sort of optimal point where you can maximize yield. And what we found was that, the maximum yields sort of could be of order 50 to 70%. This maximum yield, of course, depends on the structures that are competing with it, and the energies and entropies of those structures. And that for say a modest number of components, different components, say 100 components, if you play the game correctly, then that's how high of a yield you could get up to if you were trying to build that. Now, I'm not sure that that's impressive or not. Like, if you build ribosomes out of large numbers of components, it's not clear that a 50% yield is good enough, but that's sort of what you get out of something simple without allosteric interactions or anything like that.
So Michael, in this Big Ben exercise, did you have to tune the chemical potentials of the different species?
Right. So in this particular Big Ben exercise, we completely cheated, and we pretended that we had a box where there was only one copy of every species. And so there was no need to tune chemical potentials because, embarrassingly, there weren't any.
But you could make it work. But you had the same number of species as bricks in the Big Ben.
Yeah, it's just completely dumb and embarrassing. Of course, right? So if you did it at finite concentration, I'm going to talk about that in a second in another model problem. And tuning concentrations, as you suggest, can be a good idea, right? That is, you can get a lot of mileage out of tuning concentrations. But whether or not, what happens when you include many, finite concentrations of objects depends a lot on what binding patterns are allowed. So in this problem, the way we set it up was that we were imagining that we had colloids coated with DNA, and therefore we allowed the interactions to be isotropic. So every particle could bind to as many particles as could stick to it. And so if you did this at finite concentration, you would get like a massive aggregate because there was nothing stopping, right, more things from sticking to each other. But of course, if you really wanted to design, and you could get around that, you could put in angles and rules and things like that. I'll get to that in a little bit, but that's what we were just trying to start out simply.
Thank you.
Okay, so this is this is sort of whatever statistical mechanics. So the yield is the desired thing over everything, and everything comes from junk. Basically, there's stuff that you don't want, and there's this fact of combinatorics, which is that there's a combinatorial explosion of the denominator that basically is competing against the energy of the denominator. And the question is to somehow, basically design control variables or something to get rid of this. Now, one comment is that actually, in any system that I'm aware of, including the ones that we will talk about experimentally towards the end, there's always crosstalk. Like if you're designing interactions with strands of DNA, and so you have, the two particles have two complementary strands. Random strands, of course, still bind to these, and that still limits yield. And so in the DNA and nanotechnology business, for example, there's huge, a huge amount of work goes into sort of finding annealing protocols to be able to make things work, which are really about taming everything else. And that is hard and puts a fundamental limit on what can be built. So, okay, so getting to David's thing about polymers. So this is a calculation that Arvind Murugan did when he was a postdoc, which was that we were interested in this question of finite concentration, and Arvind basically took monomers which stuck to each other specifically, but put them in solution at finite concentration, and tried to see how the concentration matters, as David was suggesting. And what we found in this was the following. So this is an exercise where what we tried to do is we tried to design polymers which had fixed length. So for example, this point is trying to design polymers which had length 10. The way we did this was by putting a finite concentration of monomers in solution of 10 different monomers. So there was one, if it's of length 10, each one is different, and it was programmed so that it would form a monomer of length 10. Now, this one at 20 was programmed. This was 20 different monomers that were programmed to form a monomer of length 20. And so what we found was that when you compute the yield, assuming that all of the monomers are in solution at equal concentration, that the yield decays exponentially with the size of the structure that you're trying to build. And that of course occurs because of this thing I was describing in the denominator, that as you end up with longer things, there are more things that can go wrong. This was designed in a parameter regime where off-target binding was not dominant, but it still occurred at some amount, and that caused the maximum yield that you could get to plummet with the size of the structure. But what we found was that if we allowed ourselves to also tune chemical potentials to make them unequal, so that the concentrations of the different species were unequal, that we could find concentrations such that the yield was essentially independent of the structure size. And this basically is could be understood by, well, it could be understood sort of completely, but it was a sort of control knob argument that by tuning concentrations, you have precisely enough freedom to sort of cancel out the thing that's degrading you before. And so this is sort of interesting because it sort of is the sort of David's comment that, but what about finite concentration? It sort of makes you think that like there's at least a way out. But of course, in realizable experimental situations, tuning concentrations is quite difficult. Biology, tunes production and degradation machinery, presumably in a way to affect concentrations. But this is sort of another baseline theoretical result. So one other thing I want to mention, and then I think I'm not actually almost through my second 20 minutes. This might not have been a well-planned set of 20 minutes, is another thing that we sort of got interested more recently was, and this is again in a purely notional problem, is whether or not it was possible to design the kinetic barriers between states. You know, like, I mean, for example, this thing in the game of life is like beeping back and forth. And you could easily imagine building one of these if you had something which would just, you know, click back and forth between two states, right? Perhaps just jumping back and forth between two minima. And so if you imagine taking, so what I'm going to show you is, if you imagine taking one of these colloidal clusters, so these are colloidal clusters that are built out of 7 particles, and imagine that I build them. I mean, if you just put them in solution where they all have the same binding strength, they, of course, you get some yield of all of them and there's no controllability. So now what I want you to imagine is, imagine that I make this one, say, and I make it by choosing each of the 7 particles to be different, to have a different binding pattern with the others, and to use the rule that I told you, where you choose the binding rule so that it forms the thing that you want. So now what I've done is I've made it so that it will form the thing that I want. But now I still have lots of energies left, right? I just chose the energies so that it forms the thing that I want. So what we wanted to ask was, could we design kinetic barriers like that? They're, you know, if you break one bond here, it will flip from one structure to another. Can I design the height of the barrier so I can control how quickly it flips between 2 different states? And we in this case is Carl Goodrich and Ella King, sort of figured out a way of computationally, and I guess I don't really have time to discuss this, but sort of computationally designing the energy barrier of the transition states between different states in this. So these are now colored. The colors denote that they have different binding patterns, but each of these essentially interacts with each other according to some short range interaction with a well depth that you can control. And the question was that by controlling the well depth, can I sort of, you know, control at will the energy barrier between different states? So this is a reaction coordinate. This is a reaction path from one state to another. These are different structures. And, you know, what we wanted to do was to see if we could design this barrier. And basically, we sort of algorithmically found ways of doing this quite efficiently and discovered that, you know, it's possible to design quite a lot about these barriers, essentially at will, large numbers of them, to make the barrier what it wants, which seems like a useful tool for designing, for doing the sorts of things that we're talking about. And this is just a sort of a simulation of a comparison. So this is a designed, this is a structure that pops back and forth between these 2 states. And this is a Brownian dynamic simulation in which you see it popping back and forth between the 2 states. And what we did was we measured the rate. We did lots of simulations like this where we measured the rate for it popping back and forth. And then we had designed the energies of interactions to hit some target. And this just shows that we have complete control over what what the rate that it pops back and forth with. This seems like an interesting element of something that could become a machine, but right now it's just something that's popping back and forth, and at least we're able to design this barrier. So that's another sort of thing that you can do. So finally, let me say that sort of this is all sort of boring. I mean, like, we're basically designing things that assemble into controlled structures. We're designing things that can, you know, oscillate back and forth between 2 structures at different rates. But, you know, in order to get something which is actually interesting, you somehow have to input energy into the system. I mean, that's getting back to this living thing stuff, right? That's what you need to do. And we spent some amount of time exploring, in the context of simple models, how one could do that. And the instantiation that we chose, and this is a theoretical idealization, which at least, you know, experimentally could be made, I think, with colloids, but to my knowledge hasn't been done yet, which is to somehow allow the bonds to melt over a tunable timescale. So you have to make a system where you form a bond, but then that there's something else that causes the bond to melt over some timescale. That, of course, allows the input of energy. So this is expanding the rule set beyond what, you know, I talked about before, to make it so that the bonds can also melt. And in doing this, and this is work of Zorana Zarauchik, who did this when she was a postdoc, you know, we were able to sort of make self-replicating clusters. So this is an octahedron that replicates itself with a dimer. So it requires both an octahedron and a dimer. And basically what happens is that each of these, they look like they have the same color, but they don't really. Each of these has a different color that is designed so that it forms into their respective structures. And then what happens is that each color binds with its complement from the bath. And if you look on this thing at the top, you see how there are these 2 floppy bonds, and this dimer has these 2 floppy bonds. Those get together and form 4 bonds, right? And so you can see it down here. These are 4 bonds. and then if only this guy and this guy would form on top of the 4 bonds, then you would actually have an octahedron that could form if the structure melt, if the bonds that are sort of templating these reactions onto it were to melt over the appropriate time scale. And what we showed was that this isn't, I mean, this is a picture, and so it looks like there's almost nothing, but our rules for ourselves were that we had to be able to, anything like this, we were allowed to make bonds melt over some time scale, but we had to be able to program this into a computer with colloidal interactions that were specific, such that we were able to produce whatever we were looking for with high yield. And it actually turned out, again, to be remarkably difficult to do this. There were lots of kinetic traps that had to be avoided. So one kinetic trap, for example, is this one, which is that, you know, these 4 get together with these 2 get together with these 2, but then this guy on the top sticks to these 2, and then the whole process ends. And you somehow have to design the energetics of the thing so that you avoid these. But it was possible, even just us sort of playing with parameters in a computer, to get it to self-replicate. This is the sort of larger cycle that I will skip. And this is a simulation that Zorana did showing the sort of the scheme works and that there's sort of exponential growth of clusters. Another thing that we did, and this again was with Zorana, it was a sort of similar type of thing. Suppose we have melting bonds, what would happen? This is sort of the analog of one of the game of life things, the breeders actually. What you do is you start with a cluster. This is again this is a 7-particle chiral cluster, and you play the same game. That is, each particle is allowed to bind a cognate, so that you've sort of templated a structure on top of this. And then the idea is that as long as these things stick to each other, the things that bound, and then the bonds that they bound to the backbone structure melt over some time scale, then you've just templated a reaction. And basically what we were able to show was that one could use this to template forming octahedra. So this is one structure of 7 particles that could template the formation of an octahedron, which, if one set the energies correctly, one could get. But then it turned out that when we simulated it, one got octahedra, but one got like lots of other structures too that formed because of errors in the process. And you then end up with like a box full of all of these structures, each of which starts templating more structures, and you end up with an explosion of different structures that form, which is sort of reminds me a lot of this like breeder gun thing. So in any case, I've sort of told you 3 things. It was sort of silly, but they are programmed in real materials, one of which gave rise to something which was like an oscillator. It wasn't a periodic oscillator. It was tuning a transition barrier, so it would at least randomly go back and forth between two states. In order for it to be periodic, you'd have to put an energy in some way. And, the question of, just how would you do that synthetically? I showed you the sort of the structures that create more structures. And these are things that can be done with only particles that can be programmed with respect to their interactions. And you also need to be able to program the melting of bonds. And I think I was going to show you this other thing, but I think I'm going to skip this. We also, just because I'm conscious of being out of time. So that was the that was the sort of toy problem land in this. Although maybe I should be careful. And we always hoped they wouldn't be toy problems, that they would be things that could be built and done with colloids, although that seems to be, to my knowledge, hard. So that's the end of my second 20-minute section. People are still here. I'm
There's some questions coming in the chat.
Oh, cool.
Michael, can I ask you about this example when uneven concentrations allow you to stop this exponential decay?
Yep.
Do I understand correctly that in order to do that, you have to have exponentially different concentration of different components in order to compensate for exponential in energy?
That is a good question, Sasha. Exponential. So certainly you need to tune all of the concentrations differently. I actually don't think that they have to be exponentially different, but I must admit that I might be remembering wrong. But they have to be different. You're right. Your intuition is right that you're using the powers of the concentration and the partition function to essentially control the higher order terms that come up because of,
Exactly.
come up because of misbinding. But
Exactly.
Yeah, but I don't think that they have to be exponentially different.
Because I'm thinking that you need to compete with chemical potential against energy.
Yeah, so what you do, you're competing chemical potential against energy. So it's more things like this. If you're trying to create a polymer of finite length, then you need to basically make the concentration of the ends higher than those in the middle, because that way you will make it so that it's more likely to form the ends, basically, right? But it's not, orders of magnitude off. Anyway, that wasn't a very good answer. I'm sorry, but that's actually, it's a good question, and I don't remember.
Okay, so thank you.
But I think it's not exponential.
Okay, thank you.
But in any case, whether it's exponential or not, that's not to say it's useful, because if you have to tune all of the individual, and this is sort of why I got depressed about this at some point. Like, it was like, oh, that's great, we can solve this problem, but like, if you have to tune all of the parameters on their own, then that's terrible. Like, I don't know how you do that.
So Michael, I had another question in the self-replicating object in the game of life that you had on a previous slide. You said that grew quadratically. What does that mean? Quadratically in time, or the number of
quadratically in time? It's that one thing creates another thing, and that thing creates another thing. So the sort of the number of different structures grows quadratically in time.
So it's not exponential in time as would be normally the case in biology.
Well, it would be exponential if you were replicating the same thing, and then the same thing were giving rise to the same thing. But if everybody produces something different, right?
I see. Okay. Thank you.
Okay. So
there's a remark, Michael, in the questions of the chat from someone about the a mechanism for self-melting.
Oh, self-melting. Let's see. Self-replicating cluster needed. For self-replicating clusters with DNA-mediated colloidal interactions. So the DNA-mediated colloidal interactions cause things to stick, right? Like, if you put complementary strands on, then they will bind to each other, but there's nothing to make them unbind. So the scheme that I'm aware of to make them unbind was invented in the DNA nanotechnology world, in which what you do is you put in hairpins, DNA hairpins in solution to competitively interact with the strands that are bound in a way to displace them. So that of course requires spending energy, because you have to, if you have hairpins, you have to open them up in solution. They have to bind the part has to bind to the other part, which will displace them and make them melt. But it's that sort of thing was the scheme that we actually had in mind when we were sort of just playing around with this idea, and that has been, to my knowledge, it hasn't been implemented in colloids. Vinny Manoharan has actually shown that you can beautifully tune the interactions between colloids with DNA with strands in solution, but it wasn't quite this. So that's the only idea I know. But the problem is, and I guess my sort of being depressed about this at the moment, I'm now sort of depressed about all of this, is that it's great to make theoretical models. Like, there's sort of two types of theoretical models that I've discussed so far in this talk. There's the cellular automata, like von Neumann, who was making a point, right? He was showing the mathematical possibility, the mathematical consistency, right, of a set of rules. There's the sort of type of models that I was showing you in the last section, where we took like models of actual colloids. We were like, well, we're going to program, but we're going to do it with actual things, and they're going to be in the computer. And we're going to only break it in certain ways, which we need to make it more lifelike. And that's the sort of track we were on for quite a while. And I guess I'm now depressed about this. Maybe this is just the stuff of life. One always becomes depressed about things. But I'm not depressed about this but if they can't ever be instantiated in the lab, then why do I care? And so that's sort of what brought to this third part, which I'm going to start now, where we've been trying to find experimental systems and to work with just experimental groups where we can try to basically play these games on things that could be realized. And and these are the sort of my three favorite current attempts. And if anyone has other ideas, then you should say. So, but does anybody have anything else to say before I do this? Because no. Okay, I'm trying to follow the rules at least. For 20 minutes. I actually like this 3 20-minute thing. It sort of makes you organize your talk.
You're doing a great job.
Okay, I'm going to keep going because this is okay. So I'm going to tell you in sort of briefly and quickly about our 3 current efforts to instantiate these with actual materials, and these are all in flight. Like this is, do not expect, miracles, but this is the sort of what we're chasing after as we speak. So the first is magnetic handshake materials, and this is the brainchild of Chrissy Du, who's a postdoc at Harvard, and also Itai and Paul from Cornell and Ran. And these guys invented this, and they showed it to me at some point, and I was like, wow, this is amazing. This is exactly what we need. So I think I'm really excited about this as an experimental system, and I'll try to convince you of why, but then time will tell whether we can figure out what to do with this. So what's nice about this system is that it's entirely physics-based, and you'll see what I mean by this. So this was Chrissy, Itai, Paul's idea. So the idea was that what you do is you take a substrate, maybe made out of PDMS, and you print on it a dipole pattern of just magnetic dipoles. So, so this is the top view. So, positive and negative poles of a dipole. And then you use this to build panels. So now I basically can build panels which have some dipolar pattern on them. And if I take these panels and you think of them as my stickers, now basically I've just got a new way of painting stickers. Namely, what I do is I just paint patterns of magnetic dipoles in the thing. And so actually I should say, so Paul and Itai have 2 ways of instantiating this. One way of instantiating this is to do it literally with PDMS, in magnets that are sort of on the millimeter scale, in which case you end up with lots of blocks which have binding patterns on them that you can like put in a box and shake them up and see if it assembles into something. So, so the noise source in this case, that is the noise, the Langevin noise in the equations comes from just shaking, which of course it's true that it's uncorrelated, I guess, but it's problematic. But anyway, it's a nice model system upon which to test things. And then Paul and Itai also do this lithographically. So the idea is that you can sort of use nanotechnology to print patterns of magnetic dipoles on littler things and put them in solution, and that's really much more powerful. And so the thing that excites me about this system is that we can it's addressable, right? Because we can paint magnetic patterns on things. It's modelable because now we're just talking about blocks and magnets. Like we can do that. We don't forget DNA and all of that stuff. Like forget the idea of modeling, right? Of sort of hoping that you've got the off-target binding right. This is something we can think about. And you can make them reconfigurable. You can in the experiment put on magnetic fields in various ways. There's just like a ton of programmability that can happen in this. And I think especially in the lithographic system where the components can be small, there's really, it just seems like a wonderful playground that can be instantiated experimentally to sort of explore these types of ideas. And so that's what we've been working on for a while. And I guess let me just sort of show you. So this is just making the point that if you start thinking about this, so we're now going to build stickers. So you could imagine, right, 4 faces. These are 4 different types of panels with the dipoles up or down. These are the 4 different things. You can write down the formulas for what the potentials are, the interaction potentials are with these dipoles. So if you let A be the distance between subsequent dipoles, and you imagine that D is the distance between the center to center distance of these 2 things when they touch, then those are the 2 parameters that you have to deal with. And Itai and Paul and Chrissy and Ron have sort of used this in the shaking system to make all sorts of different things out of it, including with flexible components and stuff like that. But again, the one that I'm personally, and I'm not, the one I'm personally the most interested in is really the assembly out of small things with where you're like painting the interactions on the building blocks. Okay, so maybe just one slide of something. So an interesting question, which is I think sort of where we are right now on this, at least from the theoretical side, is how do you, so if you're coding college with DNA, then what do you do? So you pick DNA strands of some length, like maybe 10, and then there are 4 to the 10 different strands that you can use to coat them with. And those are the glues that you have. Now, of course, there aren't really 4 to the 10 different strands because if you take every strand in its complement, you need to make sure that you design your strand set so there's enough of an energy gap between the on-target binding and whatever the next one is, so that this is actually a useful system for self-assembly. And there's a sort of interesting question about what is the best way, sort of what is the capacity, as it were, to program this system where you can paint as many dipoles as you want, up or down, as anywhere you want, to just sort of build lock key pairs within which to start programming. And I guess so there's a paper. So we wrote a paper a couple of years ago that was sort of, I know this is Bill's thing and Bill loves mutual information. So I had to have mutual information in this talk for Bill. So here, Bill, here's mutual information. So that is, we basically tried to figure out a practical way. This was the effort of this paper, was to figure out practically how could we choose, given that locks and keys are being chosen from some set, like in this case, we're going to paint the magnets on the thing. How do we choose a set of lock key pairs that is as big as possible, minimizing crosstalk? And we sort of decided to do that in a way in the end, after sort of much futzing, by just posing an optimization problem that maximized the mutual information, where if X is the locks and Y's are the keys. So you imagine that X is one set of magnets and the keys are the complementary set of magnets. Then you can sort of compute the mutual information. You can sort of have a large set of these that you're choosing from. And what you do is you choose your set such that they maximize the mutual information. And if the binding energy is given by something that there's some on-target binding S, and the delta IJs are the errors when you bind. So if you bind something that's not supposed to bind to the right. So if you're on target, if a lock is binding with its own key, delta is 0. And so delta IJ refers to what happens when a lock isn't binding to its own key. Then what one can show is that the effective number of lock key pairs that you actually have is given by sort of this formula, where n is the number of lock key pairs that you actually have, and where e to the minus, where this is the sort of averages over the distributions of off-target bindings. And this actually is a really nice formula that just sort of follows, and you can use it to basically design, in particular, you can use it to sort of design lock key pairs. And so Chrissy has been doing this, and I just want to show you a couple of interesting things in this regard with these magnets. So this is, so imagine, for the sake of argument, we're going to start with just 1D magnets. So this is like the DNA version of magnets. So we're going to line the magnets up, right, with positive and negative in a 1D chain. And so then if you look at them from the side, right, then when they bind, they will look like this. So, and there's D is the distance between the center to center of the magnets, and A is this distance. Oops, I didn't want to show you this. So if you plot the effective number, you can sort of enumerate, so you can, for any L, for any number of magnets, so L equals 4 means there are 4 magnets, L equals 5 means there are 5 magnets, you can compute the effective number of lock key pairs one has as a function of this ratio D over A. And it turns out that actually that when D over A is 1 over the square root of 2, that that's maximized. That comes about because it turns out that that is the geometrical parameter that minimizes the interaction of this magnet with this magnet. What you're trying to do is to cancel out the second nearest neighbor interactions. And when you do that, you basically, that as a design rule, D over A is 1 over the square root of 2 does that because the dipoles are basically at, the dipolar field from this guy ends up being at 90 degrees to this guy. And so it cancels it out. And when that happens, then then that's the sort of right design choice. And so using this, Chrissy basically has sort of in her computer first built panels of these magnetic blocks. This is now designing them out of plane. So they're not all on a line, they're designed in a plane. Actually, I think I have this, I think I make the order of the slides, but that's right. So this is the, so what she did was she took 3 dipoles per face. So this is, so for each face, these are, and she optimized the dipole patterns that that you should put on each face so that the set of dipole patterns is minimally interacting from the sense that I'm talking about from crosstalk. And this is what they look like. This is what the optimized ones look like. So 0, 1, 2, and 3. This is the dipole pattern, and this is the interaction energy of the lock and key interactions between the dipole patterns. You see there's on-target binding. There's still off-target binding that you can't get rid of, but this is the one that sort of optimizes it. And then what she did was she instantiated this in her simulations that she made in HumD Blue, where she basically built faces with with the interactions that come from this, and she tried to assemble a 3x3 square. And when she did that, then this is sort of a number of simulations, and there were some errors. Some of them sort of found errors, but the yield was sort of was high. And then what happened was, so this is a plot of the yield as a function of temperature for sort of many runs of the designed strands, which are the ones which have designed interactions, versus just purely randomly chosen magnets. And you see that when you design them in the system, you can get the yield to be, higher than the random ones. This is the glassy region, so this is the region where you shouldn't look at. And then what happened is that Alice, who is a postdoc with Itai and Paul, sort of built this experiment. So she actually, in these like PDMS blocks, put these little magnetic patterns in, and hopefully this movie will play. And she see, so it basically worked. It sort of formed into the right pattern. You see the numbered blocks and the numbered things. And so I don't know. So that's a sort of simple demonstration, but it at least shows that one can design in this system. Oops, next slide. So that's one problem area that we've been working on. So a second, I'm just going to tell you 3 problem areas. That one, though, I should say, I'm really excited about that one. I just feel like that we have everything under our control, both on the simulation side and the experimental side, much more than any of these other things. And so I don't know, I think we're all somewhat hopeful, although we still have to do something interesting. So a second area that we've been working on recently is protein self-assembly, which we've been doing with David Baker and some of his students. And in this case, so David basically, as many of you know, is a magician at designing protein interfaces. He designs interfaces from scratch, so he's able to take proteins, and he uses de novo design binding interfaces, which has the property that at least as he tells it, there's minimal crosstalk of those binding interfaces with anything else, because he's designed them from scratch. And he then has lots of components, and he throws them in a jar, and he wants them to assemble into things that are useful. Like, for example, like flu vaccines. For a while, he was talking about the universal flu vaccines. But there are always assembly problems when one does this, because you design the interfaces, you put them in solution, and then you get lots of crap out that's not what you wanted. And so we started trying to figure out if one could build a sort of, this is very much of an engineering task, but a sort of computational, platform upon which one could sort of estimate what yield should be to try to help them better design what their interfaces were. Now, of course, there's a problem, which is that although David and his group are magicians at designing protein interfaces, they can't control the binding strength. They're getting better at it, but it's not like they can program in a couple KT and we can get that reliably. So there are other issues, but the thought was to somehow that if they could do that, how could one make yield predictions from a system like this? And so we have been working. We started we in this case is mainly Anisa Kuratola, who's a postdoc, has been working on this problem with. They sort of build these trimers. These are proteins that basically form into a trimer with a designed interface. And basically, I'm going to just sort of in brief, we're finally writing the paper on this. This has sort of taken us a long time. We've managed to sort of we can go from a PDB file. If you give us a PDB file of what the protein structure is, and you tell us what the binding strength, which amino acids stick and with what binding strength, then we can solve the statistical mechanic. We have sort of code that solves the statistical mechanics problem that will compute the concentration dependence of the yield. And so anyway, so that's what we figured out how to do. And we're right now in the process of trying to use it to see if we can use it to explain experiments. Actually, the reason I should say that on this slide, so it's not just David, there's a group of Vicky Wojcicki at Ohio State. So they do mass spec. of assembled proteins, and basically try to turn the measurements that David makes using these proteins into yield curves that we try to compare with. So, and actually, I'm going to skip these slides. This was a whatever. Maybe it's worth. The statistical mechanical methods we sort of have validated in simulations. I mean, I don't know, for those of you who love statistical mechanics, I guess there's a couple, at least, of people here who probably do. I mean, the trick is that you really have to compute. I mean, the rotational and vibrational partition functions are, have to be computed correctly in order to basically get the entropic part of this right. And what we finally, after much ado, it sort of looks like it should be a straightforward problem, but when you start having like arbitrary clusters and proteins and whatever, it's a bit of a nightmare. But we finally found a way to compute it effectively. So and we're iterating with experiments. We're now at the point where we're starting to iterate with experiments. I don't know, honestly, how much hope to have for this in terms of the general vague question that I've been posing here. But, on the other hand, this is a system where, in principle, one can tune binding interactions. And there are also other biological pieces that maybe one could use to go forward. So, so finally, the last sort of experiment that I want to tell you about is we've been working with Wendell and his group on this system that I told you about before with the Synapse pathway. And this is really work that was done by Mor Nizan, who was a postdoc at Harvard who just moved to Israel, where she started a faculty position. And Mor, what she did actually was so in this paper, Wendell and his team, program different ways, there are different outcomes of the Synapse pathway. So in cells contact, it modulates the expression of coherence, and they have lots of different cases where they show, many different morphologies that can occur. And the morphologies for a physics audience are sort of, I hope it's fair to say, boring. I mean, basically, if you have particles of type A and particles of type B, and A sticks to A and B sticks to B and A also sticks to B, then it's multi-component phase separation. So you either get one glob of A's surrounded by B's, or you have kinetic traps that form. And they were able to see all of this with their simulations. And what Mor did was she started by just building essentially a cellular POTS model to sort of emulate this, to try to see if she could reproduce all of the different morphologies that occurred in the experiments. And once she sort of had the thing set up, essentially calibrated so that it did do that, she started playing with different instantiations of this. And the instantiation that she ended up settling on was this one. So this is sort of constitutive A and B. When you just mix A's and B's together, you end up with like little clouds of A and amidst the C, B. But then she considered another case. This was just purely a computational dream, that when A sticks to B, you start out with A and B only weakly sticking to each other. But then when you make it that when A sticks to B, that it makes B stick more strongly to itself, so that there's actually like an allosteric effect. And she showed that when you did this in the right parameter regime in these POTS model simulations, she could get clean core shell structures. And then the nice thing is that Wendell's group is able to just program this, because they can make it so that they upregulate the expression of the B cadherin when it sticks to an A through the Synapse pathway. And when they did this is a figure which is complicated, but when they did this, then roughly, it works, so that you can sort of program it to get sort of more robust core shell structures when, in this constitutive, in the induced system than in the constitutive system, where you see that there's sort of much more heterogeneity. And this is a very sort of simple example, but it's one that that I think at least shows that this is not a crazy system to imagine trying to program. But this is clearly only the beginning of it. I mean, the interesting problem, right, is that you have cells that when they stick to each other, that depending on their chemical state, which could arise from cells excreting chemicals at different rates, or it could come from the stress state of the system. From the stress, it could make decisions to try to express, adhesion molecules, or divide, or express other morphogens. There's a whole program at play in this. And we've been, recently trying to develop computational tools on sort of model problems, but the hope is being sort of realistic model problems, in the sense that colloids are a realistic model problem, to try to understand how these programs can work, and to see whether or not there's some way of instantiating them experimentally. And that just sort of reminisces back to this picture I showed you before. So anyway, I'm not going to show you this again. I'm going to go to the next slide. I think I'm now done. So anyway, that's basically my talk. So this is a research program that we've been doing, like, aging is brutal. I guess I've been doing this for, a decade now. And I must admit that, it's not, when we started doing this, I sort of I was more hopeful than it ended up being. Like we still haven't built, in reality, anything that's a sort of example of a living synthetic material that I would, want to try to sell to anyone, as an interesting example. I mean, we can build things in our computers using the laws of physics. It's hard. It's harder than you would think. The design space is large, and there are lots of issues. We have learned a bunch of things, and right now, basically, the way that we're proceeding with this is to try to work deeply on a couple of experimental collaborations to see if we can just get better at optimizing on one hand, because one has to get better at optimizing to do this, and at the same time, somehow build something that would be interesting. And so this is my research group when we were all allowed to get together. I think that that's it. So this is the end of the talk. So anyway, I told you at the beginning that this talk was going to end on the inability to emulate biological function help. And I guess that's a good way to end it. So thank you all for your attention.